So, precision microbiota engineering for child health. Just let me get a pointer so I can also point to things. Great. Matthias is obviously going to introduce this topic in far more elegance and detail than I will be able to, because it's really his core research topic. It's something that we've got into relatively recently, but in order that we can all start from roughly the same page, at birth is really the moment when we go from being in our happy, safe, food on call the whole time, nice in utero environment, into the big wide world, which is full of, all these strange sounds and sights and smells and microbes, right? Really, really a lot of microbes. And during the first years of our life, we accumulate microbes into our microbiota. We know that various different things affect how this consortia assembles from the beginning. So things like mode of birth, things like the type of feeding. There's a very big shift that you see always at weaning of infants. So when they start to consume solid food, to the point at which you start to have a microbiome that really resembles the microbiome that we recognize of an adult human. So having this very, very dense colonization of the large intestine with something in the order of 500 different species. These are very diverse species and also relatively dynamic in different phases. And these microbes you pick up by vertical transmission. So some of them you directly colonized with from your mother, either during birth or shortly after birth, but also quite a lot of horizontal transmission. So acquiring microbes from your environment, so from other people you come into contact with. And I very much like this image here, which is genuinely horizontal transmission between definitely some cats and this toddler. Right? So we get microbes from all over the place. Anyone who has kids will recognize this as quite a common occurrence. And what we were interested in, therefore, at the beginning of this project is we know that the composition of these microbes during early life is important for many different aspects of health. So there have been links, for example, to our risk of developing allergies, our risk of developing autoimmune diseases, even neurodevelopmental issues. And how can we actually, therefore, use this resource, use this composition of the microbiota to influence health? And the challenge with all of those diseases, so things like allergy and autoimmunity and neurodevelopment, is yes, there are correlations between microbiota composition and the diseases themselves, but we know very little about the actual mechanisms that link those things together. And we therefore chose to work on two conditions where we at least have some grip on what the mechanism is that links microbiota to the disease phenotype. And those two conditions, or two groups of conditions, are inborn errors of metabolism, particularly urea cycle disorders I'm going to talk about today, which are related to the burden of ammonia produced by the microbiota, and also necrotizing enterocolitis and sepsis, which have a relationship to the overgrowth of opportunistic pathogens and also systemic spread of opportunistic pathogens from the gut. And in order to actually start microbiota engineering in these systems, we need to know what we need to achieve with this microbiota engineering. So that's what I'm going to talk about in the first part of the talk, and also how we can achieve this. So for the first question, I'm actually going to focus on these urea cycle disorders in order to understand, what is it we want to achieve with our microbiota engineering, because this is perhaps not completely as straightforward as we initially thought it would be, like everything in science. So we know quite a lot about how the microbiome influences metabolism in terms of carbon metabolism and also getting energy out of our food. So there's a lot of stuff published on this. I'm going to show one example from my group where we compare mice that have zero microbiota, so these down always in gray on the graphs, to mice with a complete microbiota that are going to be shown in blue. So they're so-called specific pathogen free. They don't have pathogens, but they do have basically everything else. And also a so-called notabiotic mouse microbiota. So this is composed of 12 bacterial strains. These are cultivable, and they've been inoculated into a previously germ-free mouse. So we know that these are the only 12 strains that are present. And the first thing that's very obvious about these mice relates to actually the digestion of their food. So if you look at the amount of energy that these mice lose in feces during a normal day, a germ-free mouse is losing almost twice as much energy, as many calories in its feces as a fully colonized mouse. And that's related to the fact it's actually also eating about 30 to 40% more than a fully colonized mouse in order to just get enough energy out of its food. And this makes sense because the mouse diet is very fiber rich. If you don't have microbes capable of breaking down that fiber and releasing fermentation products that we can use as energy, you need actually just to use more food. But it relates to actually much more than that. So it doesn't affect only the ability to get energy out of the food, it also affects type of respiratory substrates that the host is able to use during the whole day. So you can measure this very nicely using so-called metabolic cages. These allow us, from mice that are happily running around in their home cage, effectively to measure how much oxygen is being consumed by the animal and how much CO2 is being produced. And by looking at the ratio of the amount of CO2 produced to the amount of oxygen consumed, you can actually make inferences about whether the mouse is mainly metabolizing glucose, which gives you a ratio of around 1, to a mouse that would be mainly metabolizing fat, which would have a ratio of about 0.7. And you can actually follow these equations through to see why this is if you're really excited to do so. And what you find, of course, mice are nocturnal. So this is the circadian rhythm of a mouse. The mice sleep during the light cycle, during the day. They don't eat while they're sleeping, rather obviously. And so you see a drop in this respiratory exchange ratio during the day. And when the mice wake up and start eating and partying and what they do during the night, then you see this respiratory exchange ratio go up. They're using more sugar because they're getting more sugar directly from their food. But what you see if you compare this blue line, which is our fully colonized mice, to this gray line down here, which is our germ-free mice, is that actually at all times of day, the metabolism of the germ-free mice is more shifted towards fat metabolism than you see in the colonized mice. So they're relying much more on fat for energy. And then what we were very interested in, and what we still actually don't have a good explanation for, is that when we colonize mice with this 12-strain microbiota, what we seem to be able to do is to recover the function of the microbiota during the dark cycle, so when the mice are eating, but this completely fails to recover the metabolism and the energy recovery from the microbiota when the mice are sleeping. So at the moment, our best guess is that this just isn't enough energy storage during the day in the gnotobiotic colonized mice, so they have to shift back to fat metabolism during the night. Okay, but that was meant to give you some insight. We know quite a lot about the carbon metabolism, about the effects this has on the host. We know much less about what's going on with nitrogen metabolism. So here you can see your urea cycle. So we produce ammonia through the breakdown of amino acids. This is fed in to produce carbon phosphate, reacted with ornithine to produce citrulline by this enzyme called ornithine transcarbamylase, or OTC. And it's then fed into the urea cycle, eventually producing urea that is excreted via our kidneys. And actually a small part of this is also excreted via your gut. So less well known, but there is some urea directly going out through the gut. What are the bacteria doing with nitrogen? So bacteria, of course, are much more inventive on this than we are. So we do get delivery of proteins and short peptides to the large intestine, where the majority of the microbiota is. It's estimated to be around 10% of the protein we take in with our diet, as well as protein coming from things like sloughed off epithelial cells and dead cells. The first thing that the microbiota will do with that is break it down into amino acids. And actually the majority of those amino acids are probably used to make new proteins in the bacteria. This is the most efficient way for bacteria to actually use those amino acids. A small fraction of those will be either deaminated or decarboxylated and will go into energy metabolism. Of course, the deamination here releases more ammonia. The other thing the microbiota can do is they can take this urea that you're excreting into the intestine, and they can undo all of our good work of converting it into urea by converting it back into ammonia and carbon dioxide. They do this with an enzyme called urease, again, releasing quite a lot of ammonia into the gut. And then in the other direction, they can, of course, also consume ammonia. So they can take the ammonia from this urease reaction or from the deaminations, use it for de novo synthesis of amino acids, which again get incorporated into proteins. So this whole equation can go in both directions. But the general feeling is that the microbiota generates an ammonia burden for the host. And this comes from quite a few different bits of data. So one is that if you look at urea cycle intermediates, so L-glutamate and also arginine, these are elevated in mice that have a microbiota compared to mice that don't have a microbiota. One should, of course, note the total concentrations of something in a system doesn't tell you anything about the flux through the system. We need to do labeling experiments for that. But the basic data, you see higher levels of glutamate and arginine in colonized mice. This is in the liver. You see something similar in the plasma. The explanation for this is usually put onto this urease pathway. So this is thought to be the main system in the gut that is causing this urea burden. And if you look at who is capable of producing urease, you actually find this gene quite widely spread over the bacterial tree of life. So you find it in all of the proteobacteria, so alpha, beta, gamma, and epsilon proteobacteria, but also in quite some gram-positive bacteria. Intriguingly, it's non-uniform, so there will be members of the same species that do or don't produce urease, and it's quite often associated with pathogenicity. So very prominently in E. coli, the main E. coli's that produce urease are EHEC E. coli, so enterohemorrhagic E. coli's. And a demonstration that this can be actually quite an important contributor to the ammonia burden is that you can take conventional mice, so normal mouse from your animal facility, treat them with antibiotics and laxatives to try and clear out the endogenous microbiota, and then colonize them with a consortia that doesn't contain any urease genes. So they use the altered Shadler flora for this, is completely urease free. And if you do that, you can actually quite strongly suppress the amount of ammonia in the gut of these mice. So what you see is much, much less ammonia, actually up to 60 days after this intervention. And at least correlated with this, if they then come in and do a model of acute liver injury in these animals, you see much less pathology, you see better survival in the mice that have less urease producing bacteria. Caveat of this, of course, is that these bacteria are urease negative, but they also, of course, are doing all sorts of other things. So it's focusing a little bit only on the urease. We're not sure whether this is what really drives this improved survival here. Another model that's been used a lot in the field are these so-called OTC SPF ASH mice. And these are a mouse model of a very mild urea cycle disorder. They have a mutation in the most commonly affected gene, this OTC protein encoding gene, which is X-linked. It's also X-linked in mice. It's therefore the most common mutation because males carrying this mutation are hemizygous and have full penetrance disease. The mice actually have a spontaneous mutation in exon 4 of this gene, which screws up splicing of the mRNA. So what happens in the mutated mice, you can see this is a good old classic northern blot here. In the wild type mice, we have 2 major bands. In the mutated mice, we have all kinds of bands coming up with actually very little correctly spliced OTC, but just enough that the mice survive, and actually they do rather okay without protein challenge. What has been done in this model is actually not to take out the urease, but to see if you can affect the equation in the other direction. So taking a microbe that can sequester, actually accumulate arginine, and using this to sequester ammonia out of the system. And this is actually results from very extreme experiments done by these authors where they took the OTC mice, they challenged them with an extremely high protein diet. So it's a 70% protein diet. Either they were untreated here, in which case you see the blood ammonia go up, and there was actually a lot of lethality in this untreated group, or they were treated with this symbiotic, so this E. coli nissel accumulating arginine. And in this case, they managed, or at least many of the mice managed to maintain their blood ammonia level at a similar level to what was seen with only the chow. So this looked quite promising that you can actually sink ammonia into the gut even in these very, very heavy protein challenges. There's a lot of caveats in the discussion of this because potentially you just have a lot of delivery of protein into the large intestine when you give such a high protein diet. But we wanted to go a little bit further and do this in a cleaner way. So we have re-derived this mouse model into fully germ-free conditions. So they have no microbiota at all. This was done by the animal facility, but really managed by Elisa Capio, and the analysis of these mice has also been supported by Jens Scheidecker, who's our master's student at the moment, with support from the group of Johannes Hebeli at the KISP, who was originally running this model. And the results that we got were actually quite surprising. So we started to notice this when we started breeding the mice. Our initial hypothesis here was going to be that the germ-free mice should do much better because the microbiota is considered a source of ammonia. and therefore the germ-free mice should be pretty healthy. And really what we see was the opposite. So we were having a lot of trouble breeding the mice under germ-free conditions. We had to reduce the breeding protein diet percentage in order to get litters of mice that contained OTC males. And actually now Elisa has done the full comparison in metabolic cages. There's a lot of data. I'm just going to show you a very small amount of it here. But we see similar failures of splicing in the SPF and the germ-free mice in this model. And we see that the germ-free mice do much worse, actually, even in a very, very mild protein challenge. So we're going from our low protein breeding diet that we specifically use from these mice to a standard breeding diet, which is 18% protein. And actually 3 out of, here we have 5 OTC mice in germ-free conditions, lost very significant amounts of weight, sufficient amounts of weight. We actually had to euthanize the animals or showed disease symptoms, whereas the SPF mice did rather okay. And actually, if you look at what's going on at the level of the overall metabolism of these mice, again, what you see is that the SPF OTC animals look pretty similar to the wild types. They have a nice circadian rhythm in the RER. The germ-free mice that have this OTC mutation were basically eating continuously. So they were eating small amounts of food throughout the day and night. They completely lost circadian rhythm. And you see this reflected actually in an inverted RER in the germ-free OTC SPF ASH mice. So what this seems to suggest is that actually, yes, clearly you can have an effect of urease releasing a lot of ammonia and producing an ammonia burden for the host. But in a lot of the cases, it looks, at least with our SPF microbiota, that this is really acting as a very good ammonia sink for the host. So it's actually really helping the host to deal with the ammonia burden. So the interpretation of this for us is that we're not doing something completely ridiculous. It is actually an interesting model where we really can use microbiota engineering to influence the disease outcome, hopefully also in human patients. And what we really want to do is to be able to suppress the urease producing bacteria, as well as to boost the ammonia consumers in the gut. And once we've got to this stage where we know what we want to do, of course, the next stage is to be able to actually do it. And we hope that this is where our knowledge of being able to manipulate the immune system in the gut becomes useful. Because what we have done to a large extent until now, if we want to change the composition of a microbiota, has been relatively high-level guessing. So we would, for example, take one of our known probiotic strains and swallow it and hope that it will engraft in the gut, even though a lot of our probiotic strains are not actually gut bacteria, right? They're very typically from fermented food rather than from gut bacteria. It's a little bit like taking your rainforest and planting your edelweiss and hoping like crazy that it's going to grow, and normally it doesn't. At the other end of the scale, we have fecal microbiota transplants, right? This has been very successful. So it's been very successful in very specific cases. Clostridioides difficile colitis is the main one of these still, where there's been massive progress with fecal microbiota transplantation. And this is a very different situation because normally that disease arises after very heavy antibiotic treatment. And that means you have effectively a massive open niche in the gut. You're taking real gut bacteria from a real fecal sample and basically planting them onto the fertile soil of the C. diff gut, where they can grow and outcompete the C. diff and the patient gets better. But in all of these other cases, it's been much more complicated, right? You can IBD, there've been many, many trials with fecal transplantation or with probiotics, all with very uninspiring outcomes. So it's quite difficult to work out who is going to respond and what's going to engraft and what's not going to engraft. It's a little bit too much guessing for this to be clinically useful. So at this point, I'm going to shift from talking about the inborn areas of metabolism, because there we've done less work on this, to the necrotizing enterocolitis and NEC, where we know what we actually want to target, which at the moment are actually multi-drug resistant E. coli's. And the data that this is potentially useful comes from some quite nice clinical studies that have been done in NEC. We've also got our own study running here that has got around 80 patients involved in it now. But what they've been able to demonstrate, for example, is that actually in babies that develop necrotizing enterocolitis, what you start to see is an increased abundance of Enterobacteriaceae that are not coated with maternal antibodies. So they seem to escape the maternal antibody response. So what we think would be helpful and useful in particularly these very premature babies who are at a very high risk of progressing to NEC or to neonatal sepsis is to decrease the amount of E. coli transmission from either the mother to the baby or the environment to the baby, and also to improve the delivery of E. coli-specific IgA to these very susceptible neonates. And the reason that we think this is useful is that we know that secretory antibodies can be used to replace one strain with another in the gut. And this is something my group has published quite a lot on, so I'm going to go over it rather superficially. But the work I'm going to present is from Verena Lensch, who was a PhD student in my group. She defended last week. And also Louise Larson, a PhD student shared between my group and Mederic Diard's group in Basel, on using different combinations of things to change gut colonization. So the way that we do this is we design an oral vaccine. We do this rather rationally, trying to get cocktails of antigens that it's difficult for the bacteria to escape from. We call these evolutionary trap vaccines. These are basically inactivated bacteria. When you swallow them, you induce a specific IgA response in the gut. If you happen to be the patient, that's great. If you are the mother of the patient, you produce these responses in the gut, but also in breast milk. And this can be therefore transferred to the babies via breastfeeding. What these secretory antibodies then are capable of doing is sticking bacteria into these big clumps, which initially seemed not very useful, but actually anybody who's dealt with small children knows that one of the good ways to get them all to go in the same direction is to get them to hold hands. And a very similar process operates in your intestine. So if you clump these bacteria together into big aggregates, they flow much faster with the gut content and actually get cleared faster from the gut lumen. This gives them a selective disadvantage, which generates then a niche that you can introduce your favored bacteria into, and it can grow up and actually outcompete the microbe that you're trying to get rid of. And we've demonstrated this for salmonella colonization in the mouse non-typhoidal salmonellosis model. So here, if you have just the naive animal, you colonize with salmonella permanently over our 10 days of infection at very high levels. If we use either vaccination alone or a niche competitor alone, we do suppress the levels of salmonella. But if we do this combined, we can actually get sterilizing clearance of pathogenic salmonella within 9 to 10 days of infection. So we have proof of concept that it can work. And that's great for Salmonella typhimurium and also for non-encapsulated E. coli, because for all of these strains, we know that the main protective antigen on the surface is this O antigen, this glycan that's linked to the lipid A in the outer membrane. And this is nicely recognized by antibodies. It's quite easy to induce immune responses against this. It nicely cross-links and we get these nice clumps and out competition. The challenge is that a lot of the E. coli that are driving nasty infections, so the invasive infections, the sepsis, the meningitis cases, are capsulated E. coli. So they produce these big polysaccharide capsules, and these basically block access of antibodies to the immunogenic surface structures. And to make the problem more challenging, time for the next lecture, actually a large fraction of the E. coli that are disease-causing have either the K1 or the K5 capsule. And K1 is a polysialic acid that is identical to a polysialic acid that our own neurons produce. K5 is identical effectively to heparazan. So both of these are self-antigens. We have self-tolerance against these structures. So you cannot vaccinate against them. Even if we could, you probably don't want to. So here, vaccination becomes not the right way to go to deal with these E. coli. And this is where actually Mederic Diard's group has been doing very nice work using, instead of antibodies, phage. So what they do is they select phage out of wastewater that can specifically bind and kill the K1 E. coli. And they select these phage in such a way that the outcome of this is that you actually select for capsuleless mutants. So we're not trying to get complete clearance of the E. coli by the phage. We're just trying to select for E. coli that no longer produce the capsule. And this is effective in vitro. It's also very nicely effective in vitro. So you can show this with a mouse colonized with a K1 E. coli. You come in with your phage and you look over time at the phenotype of the E. coli coming out of these mice, and you find that the phage infected animals actually are switching to have basically 100% of the E. coli present in the gut, not producing the K1 capsule anymore. And this is interesting because the K1 capsule is also a virulence factor. So when you lose it, we expect these strains to be per se less virulent, but these strains also of them become susceptible to things like abnormal vaccines because they're no longer encapsulated, or to things like other drugs and treatments that would otherwise be blocked by the capsule. Okay, so I'm pretty much out of time. I can just do a short summary of sort of where I think this whole field is going. So at the moment, we've been working a lot with salmonella. You can see our pigs here now getting salmonella vaccines. So we're starting to translate the concepts of salmonella niche competition into livestock rearing with the idea to actually try and eliminate salmonella from initially pig farming. We're also now going to see if this works also in chickens. And also in the next, hopefully 3 to 5 years, we're really hoping we can start to actually do a first in human trial with one of these oral vaccines in order to check at least for safety and immunogenicity. Within the next 10 years, I hope we can actually get these strategies far enough that we can really intervene in diseases where we know what the microbiota is doing. So these inborn errors of metabolism and also neonatal sepsis. And beyond that, what I hope is that the techniques that we're going to develop to be able to rationally modify the microbiome will start to be useful for these more complex multifactorial diseases, such as food allergies, such as autoimmunity. So with that, I acknowledge the huge consortium that is our BRCCH project. So you see we have 19 PIs involved across ETH Zurich, the University of Basel, the Kinderspital Zurich, the Universitätsspital Zurich, as well as the PSI, as well as many team members from all of their groups. I've only shown a tiny fraction of what's going on in the consortium today. The rest of the people on this